Good morning everyone. Good morning. Good morning. Good morning all. Okay, good morning all. I think everyone is taking rest today because of Sunday. Okay. Okay, so friends, in the last class, we were discussing about the plant hormones. We have started discussing about the plant hormones, and if all of you quickly recall, we started discussing the hormone auxin first. Whenever we started discussing about the hormone auxin first, okay, join. Good morning, all. Whenever we are started discussing about the hormone auxin, friends, we started initially the discovery of auxin. Good morning everyone, please continue. We understand how the auxin get discovered and we understand the experiments of the Darwin and CSIRNET. Okay, so friends, listen to me very carefully. Good morning all. There are different type of auxins are there, friends. We will be calling them as the natural auxins. We will call them synthetic auxins. We call them free auxins. At the same time, friends, we will be calling it as the bound auxins. Good morning everyone. Please continue. So friends, there is another type of auxin also there, friends. We generally call it as antioxin. Everyone pay attention, friend. Antioxins. Okay, listen to me very carefully. Let's try to understand one by one briefly. Natural auxins. So friends, how can I define the word natural auxins? Natural auxins are those type of auxins, friends, which are synthesized in the plant body naturally. So that's why those kind of auxins we call it as the natural auxins. So what is the best example, friends? IAA and IBA. So I want all of you has to remember one important statement is that, friends, even though many number of natural auxins are present, listen friends, even though many number of natural auxins are present in the plant body, the most commonly occurred natural auxin is the all the time IAA only. Don't forget the statement. So that is what do we call the natural auxins. Then friends, what do we call it as the synthetic auxins? Synthetic auxins basically man-made auxins. What are the best examples of the synthetic auxins or man-made auxins? I want every one of you remember the all examples, friends. 2,4 a dichlorophenoxyacetic acid. Good morning all. 2,4 dichlorophenoxyacetic acid. At the same time, friends, 2,4,5 trichlorophenoxyacetic acid. These are all what, friends? The best type of the synthetic or man-made auxins. I want all of you to remember one more important point. That is, friends, these two actually acting like what, friends? These two are actually acting like the weedicides. So don't forget the statement. Now friends, listen to me very carefully and kindly don't get confused with the free auxin with the bound auxin. See friends, when you can called a auxin is a free auxin. Listen to me carefully. When you can called a auxin is a free auxin. Whenever you can isolate a particular auxin without any solvents, without utilization of the solvents, if you don't use any solvent and if you isolate the auxin, that auxin you can call it as free auxin. So friends, all of you listen my words carefully. So friends, a free auxin may be synthetic auxin, a free auxin may be natural auxin also. Whatever the auxin it may be, doesn't matter, friends. If I want to isolate the auxin, if I don't use any organic solvent, that is what exactly we call, friends, free solvent. Now friends, what do you mean by the bound auxin? In order to isolate the particular auxin, if I use organic solvent compulsory, then what do you call it friends? We called it as the bound auxin. Right. So bound auxin also a free auxin. A bound auxin is also a natural auxin. Friends, pay attention. I will cover every single point from every single hormone. So pay attention. Antioxins. Friends, what are antioxins? Name itself indicating na, which are against the auxin. Which are against the auxin, means which shows opposite action to the auxin or which inhibit the auxin activity, we can call friends antioxin. So friends, antioxin may be anything. I want all of you to remember. So you know, we used to heard that one hormone can inhibit the growth and development of the another hormone. Of course true. So what is the statement you have to remember? All the inhibitors, we can call it as what friends? Antioxin. So even another hormone also can act like the antioxin, or another hormone also can act like a inhibitor molecule. Don't forget this friends. I want all of you to remember most amount of the information. Good morning everyone, please continue. We are continuing the hormones. There is the first hormone, auxin. Friends, I want you to remember most of the information. In this all hormones, the only thing you people has to focus is the information with respect to the hormones. More amount of information, and the best way to remember all the hormones is comparing one hormone with the another. I will tell you how to compare and how to remember for the long time. Now friends, focus. Site of synthesis. All of you pay attention. Shoot apical meristems are the major sites of the synthesis of auxin. And friends, all of you keep it in your mind. When you heard the name, everyone listen to me. When you heard the name of auxin, what is the common statement that I want you people to remember here? The common statement you have to remember is auxin is the rooting hormone. What kind of hormone auxin is? Auxin is the rooting hormone, which means, my friends, auxin is going to show its major action on the root, but it doesn't mean that it is synthesized in the root. No, auxin is synthesized in the shoot. From the shoot, it will travel to the root, and there it starts showing its mechanism. One more important and interesting point is what, friends? I have told you very clearly that site of production and site of action is entirely different. Same way, my friends, auxin is going to be biosynthesized in the shoot, some area, and going to show their action on the root. It is the other different area. Don't forget the statement. Now, when it comes to the biosynthetic pathways of the auxin, friends, is there any necessity to remember this particular structures or of this pathway? Not at all. You do not require to remember any kind of a structure. Kindly don't worry at all. And once again, we can get a direct question, friends. What is the name of the amino acid? This is important, friends. What is the name of the amino acid that is involved in the biosynthesis of auxin? I want all of you remember, friends. That is what do we call tryptophan. Tryptophan, which is an aromatic amino acid, it is acting as a precursor for the biosynthesis of this particular auxin. And friends, what are the major pathways that are involved in the biosynthesis of auxin? There are many pathways are there, friends. We can call it IPA. IPA means, friends, indole 3-pyruvic acid pathway. Always remember. Then friends, IAN pathway, indole acetonitrile pathway. What was that? Indole acetonitrile pathway. And next one we call, friends, IAM pathway, indole acetamide pathway. Next one, friends, we can call the TAM pathway, tryptamine pathway. So I'll quickly look in summarize. IPA is the indole 3-pyruvic acid pathway. IAN is the indole acetonitrile pathway. IAM is the indole acetamide pathway. TAM is the tryptamine pathway. So friends, even though there is a presence of many pathways, the plant always uses only IPA pathway as their major pathway for the biosynthesis of what, friends? Auxin. How? All of you pay attention. This particular tryptophan undergo the IPA pathway, and ultimately, friends, it is going to give rise to the auxin. Now all of you look into this. This is the major principal pathway I want you people to remember. These are all not required. These are all occurring in the bacteria. Anyway, friends, it's very easy. As I said before, you people no need to remember the structure. What I want you to remember is the schematic presentation. I mean to say, all the steps involved you have to remember. Look into the first step. First step is what, friends? This is the amino acid tryptophan. Everyone pay attention. Amino acid tryptophan can be treated by an important enzyme. What is that particular enzyme called as? We call it as a tryptophan transaminase enzyme. Upon treating with the tryptophan transaminase enzyme, what the transaminase enzyme will do? It remove the amine group. Very simple point. Upon removing the amine group, friends, tryptophan converted into indole 3 pyruvic acid. All of you listen my statements carefully. The stable product, all of you listen, the stable product or the intermediate product produced is the 3-carbon containing compound. That's why, my friends, we will be calling it as the indole 3 pyruvic acid. Now all of you listen to me carefully. Now this indole 3 pyruvic acid can be treated by an enzyme called as decarboxylase enzyme. Friends, transaminase enzyme transfer the amine group. IPA decarboxylase enzyme remove the carboxyl group. Friends, upon removing the carboxyl group, indole 3 pyruvic acid is basically converted into indole 3 acetaldehyde. So friends, this particular indole 3 acetaldehyde can be treated by another enzyme called as indole 3 acetaldehyde dehydrogenase. Ultimately, friends, they are going to give rise to the indole 3 acetic acid, which is IAA. So friends, I'll quickly summarize the pathway. All of you pay attention. Come on, be active. So tryptophan can be treated by an enzyme called as tryptophan transaminase enzyme. Now this tryptophan transaminase enzyme transfer the amine group. Upon transferring the amine group, tryptophan converted into indole 3 pyruvic acid. Now indole 3 pyruvic acid can be treated by an enzyme called as IPA decarboxylase, which remove the carboxyl group. Upon removing the carboxyl group, it is converted into acetaldehyde. Upon treating with the dehydrogenase enzyme, ultimately it is going to give rise to what, friends? Indole 3 acetic acid. So friends, now look into the picture of a transport of auxin. So before going into the functions of the auxin, we have to know compulsory the transport of auxin. Friends, all of you listen to me very carefully. As we have already discussed, auxin is actually biosynthesized where? Auxin is biosynthesized in the shoot, and it will travel to the root, and it is going to show their mechanism in the root. Correct, na? So all of you listen my statements very carefully to understand. What to understand? To understand how does the auxin starts moving from the shoot to root. How does the auxin starts moving from the shoot to root? See what the plant physiologist does, friends. All of you pay attention. Friends, they have taken the they have taken the coleoptile. This is the coleoptile. Friends, all of you listen. They cut the middle part of the coleoptile. This is the upper part, right? Friends, upper part we call it as epicotyl, and middle part, what do you call, friends? Hypocotyl. What the physiologist does, they cut the middle part, that is the hypocotyl. And you know, friends, every one of you look into the diagram very carefully, friends. They kept this particular hypocotyl between the two agar blocks. So there is a upper agar block and lower agar block. All of you understand, friends, this particular upper agar block is completely loaded with the auxin. What, friends? This upper agar block is loaded with what? Auxin, in which the segment translocated to the lower block. Very, very important. You only try to understand in the upper agar block there is a auxin is there. After sometimes you only listen, after some time the dark blue color one converted into light blue color one and the white color one converted into light blue color one. Yes or no? Of course. So what is the statement I can conclude here? The statement I can conclude here is that this particular auxin starts moving from a shoot to the root. Auxin start moving from shoot to the root. So always remember that. Now all of you listen to me carefully. They have done the same experiment, friends, but this time what they have done, they have inverted the coleoptile. So you only look into this diagram. They have inverted the coleoptile, friends. They cut the hypocotyl. Whatever the hypocotyl that they cut, they kept in the they kept between the two agar blocks. Same thing, friends. Upper agar block is loaded with what? Auxin. Now you only try to understand after certain time there is a no change in the color. What, my friends? After certain time there is a no change in the color. So what is the unique statement you people has to remember here? Whatever the auxin that is present in the root, it never ever move to the shoot. This cannot happen. Auxin only move from shoot to the root. My friends, this unidirectional transport of auxin. Listen to me, friends. This unidirectional of transport of auxin, that is from shoot to the root, that kind of auxin transport only, friends, we will be calling it as the polar auxin transport from shoot to the tip of the root in a single direction without any obstruction. That is what exactly, friends, unidirectional transport or polar transport. Okay, friends, right now what we studied, we understand different things like what is hormone auxin, the discovery of auxin we understand, the types of auxins we understand where the auxin is getting biosynthesized, we understand biosynthetic pathway, we understand the transport of auxin, and the most important part we people has to read and remember is the functions of auxin. Why these functions of auxin is very important? 99% the questions will appear in our CSIR exam from the functions as well as the signaling. These only very important, friends. Okay, everyone pay attention. What is the function of auxin? All of you listen. Auxin is the hormone, friends, which stimulates the rooting. What do you mean by the word which stimulates the rooting? Root initiation on stem cutting and lateral root development. Friends, I will be explaining the every single point. Just pay attention and try to understand what exactly I am talking here. So let's consider this is the function 1. It stimulates rooting. So all of you look into the diagram very carefully, friends. So what is happening? Just imagine this is the shoot. So where auxin is present, my friends? Auxin is present in the shoot, correct na? So from the shoot, where does the auxin is traveling, friends? Auxin is traveling to the root. Once auxin travels to the root, it start involved in growing of the lateral branches. You can see, right? Lateral roots, I'm sorry. You know, right? Primary root, secondary root, tertiary root. These are all what, my friends? Different type of roots, right? So friends, the auxin which is present in the tip of the shoot, they travel to the root and involved in the development of the lateral roots. So that is the first and most important function of the root, of the auxin. And friends, consider this as the second point. What is that point, friends? It promotes apical dominance. It is really important function, friends. It promotes apical dominance. All of you pay attention, understand what is this particular apical dominance. Everyone pay attention, friends. This is what here auxin is present, correct na? At the tip of the shoot auxin is present. Everyone listen to me very carefully. Listen, friends, whenever auxin is present at the tip of the shoot, listen to me carefully. Whenever auxin is present at the tip of the shoot, it never allow, what my friends, it never allow the growth of the lateral branches. It never allow the growth of the lateral branches. This is what, friends, exactly we can call promoting the apical dominance. Listen my statements very carefully, friends. Promoting the apical dominance means inhibiting the lateral branches. So when the auxin is present at the tip of the shoot, it never allow the growth of lateral branches. But my friends, please listen my statements very, very carefully. I have used a word that auxin is the hormone which is promoting the apical dominance. But friends, cytokinin is the hormone. I want every one of you listen to me. Cytokinin is the hormone, friends, which is basically inhibits the apical dominance. What, friends? Cytokinin is the hormone which is basically involved in inhibiting the apical dominance. So friends, every one of you pay utmost attention. So whenever cytokinin is present here, what is happening, friends? Whenever there is a presence of the cytokinin, cytokinin inhibit the apical dominance, which means cytokinin will allow the growth of a lateral branches. So friends, once again listen to me. Auxin promote the apical dominance, which means that it inhibit the growth of lateral branches. Cytokinin will do the opposite function. That is, friends, it inhibit the apical dominance, which means it will allow the growth of the lateral branches. Very, very simple point it is. Now friends, think and use the brain and understand what I am talking. How can I use, listen to me, friends, how can we use this particular apical dominance in case of applications, or how can we apply this particular apical dominance? Do we have any kind of a specific application? Yes, I hope you all read. There is an important technique, my friends. We call it as a bonsai technique. All of you listen my statements very carefully. Suppose example, if I want to purchase a mango plant, normal mango plant or guava plant, it is hardly 500 to 1000 rupees. But the same mango plant, guava plant, if I want to purchase, which is developed by the bonsai technique, friends, it is more than 10,000 rupees. Why it is that much costly, friends? Every student listen to me. In the bonsai technique, what they will do, you know, friends, once the plant reaches a certain height, what my friends, once the plant reaches the certain height, what they will do, they will cut the tip. Simply they will cut the tip. My friends, whenever they cut the tip, immediately what is going to happen, friends? This particular thing can be released out. There is a no tip. Whenever there is a no tip, do you think a height can be grown? Height never increases. Only increases is what? Girth. Only branches can be increased. That's why, friends, bonsai developed plants we can grow anywhere. We can keep it in the house, in the middle of the house also. In any room of the house, we can keep this particular bonsai developed plants, because height is restricted and branches are just increasing. Girth is increasing. That is what, friends, bonsai technique. So please kindly don't forget this particular second function. Of course, undoubtedly every function is important. Stimulates the rooting and it promotes the apical dominance. And friends, all of you listen to me very carefully. The third point, it stimulates cell elongation, at the same time, friends, cell division. all of you listen, how it is going to elongate the cell and how it is involved in the cell division, we will read in the next slide very elaboratively with the help of a diagram without any confusion. Now currently look into this fourth and fifth points. So friends, this is really interesting point. Fourth and fifth points are really interesting points. Look into this. The fourth point says that, all of you pay attention, auxin is the hormone. What is the auxin hormone is doing? It is preventing the abscission. What it is preventing, friends? It is preventing the abscission of the young leaf and young fruit. Never ever forget this particular statement at all. Friends, listen to me. Listen my words carefully. In the plant body, young leaf and young fruit never ever fall down on their own until if there is a hormonal imbalance or until there is a diseased condition. Then only leaf and fruits will fall when they are in the younger stage also. But what is the unique statement I want you people to remember, friends? Auxin prevents the abscission. What, my friends? Auxin prevent the abscission of the young leaf and young fruit. Which means if the auxin is present in the plant body, it never allow any young leaf and young fruit not to fall on their own in the plant body. But see friends, the really interesting point. I only said that auxin is preventing the abscission of young leaf and young fruit. When the young leaf become matured leaf, when the young fruit become matured fruit, which we can call old leaf and old fruit. And my friends, surprisingly, don't forget, auxin only stimulate the abscission, means falling down. Auxin only stimulate the abscission of the old leaf as well as the old fruit. Friends, please kindly don't forget that. So what are the statements you have to remember? Auxin stimulates the abscission of the old leaf also. Auxin stimulates the abscission of old fruit. But auxin never ever stimulates. Instead of stimulating, it will just prevent the abscission of the young leaf and young fruit. Don't forget the statements at all, and easy statements. Then the sixth statement, friends, a very common statement. Auxin is the hormone which induces what, friends? Parthenocarpy. Where auxin is going to induce the parthenocarpy? In tomatoes also, my friends. Even in the strawberry also. What auxin will do? Auxin induces the parthenocarpy. What is parthenocarpy? Development of a seedless fruits. Correct, na? How we will get the seedless fruits, friends? Seedless means what? Here what exactly the parthenocarpy represents? Without fertilization. In a simple line, without fertilization, what is happening, friends? This particular seedless fruits can be produced. And friends, the last and a very important point, it promotes auxin. Auxin will promote flowering as well as a femaleness. Femaleness in what flowers, friends? Femaleness in the dioecious flower. 90% of the students, they will forgot. They will think dioecious means bi. That is absolutely wrong point. Dioecious means what, friends? Either male or female. So which means dioecious means what, friends? Monosexual. Dioecious means, friends, monosexual. It promotes flowering as well as the femaleness in the dioecious flower. Listen my statements carefully. Whenever auxin is present in the plant body, it will always stimulate the development of the female flowers when compared to the male. That is the major functions of auxin. So friends, let me quickly summarize. See, hormone is one of the most important topics, friends. Compulsory we get the questions. I hope you all remember the February 2022 paper. We got 5 part B questions and 5 part C questions from this particular plant physiology. Then imagine 10 marks here and 20 marks here. Imagine how important this chapter is. So we should not leave any kind of a topic here. Okay, I'll quickly summarize. Look into this. Auxin is the hormone, stimulates the rooting, especially that plays a key role in the lateral root development. Next friends, auxin is going to stimulate the cell elongation also, as well as my friends it is involved in the cell division also. Don't forget that. At the same time friends, auxin is going to prevent abscission of the young leaf also friends, abscission of the young fruit also. Means it never allow the young leaf and young fruit not to fall down. And I told you very clearly, it is preventing the young leaf and young fruit to fall down. But my friends, auxin is basically stimulate the falling down of the old leaf as well as my friends old fruits. Don't forget this. And another unique statement, auxin is basically induces, what it is inducing? It is inducing parthenocarpy. Where it is inducing the parthenocarpy? In tomatoes also friends, even in the strawberry also. Don't forget this. And just we discussed, it promotes the flowering as well as femaleness in the dioecious flower. So in the monosexual flowers, it is majorly involved in the development of a female characteristics. And it promotes apical dominance. What is apical dominance? Which inhibits the growth of lateral branches. As I told you, I will highlight this particular second point very elaboratively and I'll explain you. Now listen, how auxin is basically involved in the cell elongation. My friends, please kindly don't confuse. Listen to me very carefully for 10 minutes continuously. Listen to me carefully for 10 minutes. Don't confuse at all and listen to me carefully. There are two hypotheses were proposed. Why the two hypotheses were proposed? To explain. What to explain? How does auxin hormone is involved in the cell elongation? To explain the role of auxin in the cell elongation, there were two hypotheses were proposed. Friends, one hypothesis we will be calling it as friends activation hypothesis, and another hypothesis friends we will be calling it this as the synthesis hypothesis. Now all of you pay attention friends. First let me explain you activation hypothesis. Now everyone pay attention friends. This is the cell wall and this is the cell membrane, we can see that. And friends, the space between the cell wall and the cell membrane, what do you call that particular space? We can call the cell wall space. All of you look into the diagram very carefully. I'll explain twice. On the cell wall, on the cell membrane, there are H plus ATPase pumps are present. You all commonly know, right friends, what is the function of this particular H plus ATPase pumps? By ATP hydrolysis, they pump the protons into the cell wall space. That is fine. But my friends, listen to me. Actually, this particular H plus ATPases which are present on the plasma membrane, they are extremely inactive. What my friends, they are extremely inactive. Do not confuse this particular statements friends. So IAA command associated with this particular receptor. What is the name of the receptor? We can call ABP1. You people always remember the short names only. ABP1 means auxin binding protein 1. IAA will come and attaches to the ABP1. The moment IAA will come and attaches to the ABP1, what is happening my friends? All of you pay attention. Now this auxin immediately activate the H plus ATPase pumps. Understand to me carefully. So far friends, all these particular H plus ATPase pumps are inactive completely. Now what I am doing friends, I am basically activating this particular H plus ATPase pumps. How? With the help of auxin. Now all of you listen friends, when the auxin will come and activate the H plus ATPase pump, by the hydrolysis of ATP, they pump the protons into the cell wall space. Now every student listen. Now the cell wall space become what friends? Acidic. Now everyone listen to me carefully friends. When the cell wall space become acidic, immediately my friends, there will be an activation of an important protein. What is that particular important protein that is getting activated? My friends, that important protein only we can call the expansion protein. Please kindly don't forget that. What do you call it that, friends? We call it as the expansin protein. And friends, all of you listen to me very carefully. What is this expansin protein, or how expansin protein basically elongate the cell? Because our ultimate goal is this. We have to understand how the auxin is involved in the cell elongation. So auxin activated the expansin, and you all know, right, what does the cellulose is made up of? Cell wall is made up of cellulose. And friends, what does this particular cellulose is made up of? We all commonly know that, friends, cellulose is made up of cellulose microfibrils. Correct, right? There are many cellulose microfibrils come close together, they can actually give rise to the cellulose. I am drawing very loosely, friends. Actually they are very tightly associated with each other. Now all of you look into me very clearly. Actually, friends, listen to me. Cellulose microfibrils are associated like this. Very tightly they are associated. Very, very tightly. But you know, my friends, what is this expansin? Please listen to me. What is this expansin will do? Expansin penetrate. What, my friends? Expansin penetrate into the cellulose microfibrils. It will loosen the cellulose microfibrils. All of you listen. Expansin penetrated into the cellulose microfibrils. My friends, this particular expansin loosen the cellulose microfibrils. The moment cellulose microfibrils become loosen, what is happening, friends? Abundant amount of water molecules enter inside. Once the water molecule enter inside, very simple, it will cause the turgidity. My friends, because of the turgidity, cell is getting elongated. That's a simplified hypothesis, friends, which we call it as the activation hypothesis. I am explaining for all of you once again. Everyone pay utmost attention. What is happening first? Auxin, that is IAA, will come and associated with the auxin binding protein 1 receptor. Immediately what is going to happen, friends? This particular auxin will go and they activate the H plus ATPase pumps. Now this particular H plus ATPase pump immediately pump the protons into the cell wall space, and my friends, cell wall space become acidic. In the acidic environment, there is an activation of protein called as expansin. Now expansin, what it will do? It will cause the cellulose microfibrils to get elongated, or I mean to say, it allow the cellulose microfibrils to become loosen. The moment cellulose microfibrils become loosen, it will allow the more amount of the water enters inside. Such a simple step it is. This is what do you call, my friends, activation hypothesis. You can ask me, sir, why do you call it as the activation hypothesis? We called it as the activation hypothesis because whatever the H plus ATPase pumps that are present on the membrane, they are not active, my friends. That's why we are activating them. That's why the name of the pathway is activation hypothesis. So friends, the statement is very clear. Without H plus ATPase pumps, we can't synthesize the expansin. Without expansin, the cell cannot be elongated. It's highly impossible. Certain times, you know, friends, the H plus ATPases are absent. In this particular condition, you all can observe one thing. That is, H plus ATPase pumps are present. They are expressed on the membrane. But the only point is that, my friends, they are inactive. That's all. But in certain cases, you know, H plus ATPase pumps are completely absent. Then what we have to do? Listen to me. Then we have to synthesize the H plus ATPase pumps. Once we synthesize, we have to express on the membrane. Then we have to activate them. Then only they will pump the protons. Now all of you listen. How can we synthesize this particular H plus ATPase pumps? Now all of you listen to me, friends. Now IAA auxin will come and associated with the auxin binding protein 1. All of you pay utmost attention. Auxin will come and associated with the auxin binding protein 1, which is a receptor, where the receptor is present directly in the cytosol only. Now friends, listen to me. Then auxin will activate the secondary messengers. Secondary messengers enter into the nucleus. And friends, once they enter into the nucleus, they are going to activate. What they are going to activate, friends? They are going to activate the H plus ATPase gene. What they are going to activate, friends? H plus ATPase gene. Now all the students pay attention. This particular H plus ATPase gene only, right? It undergo the transcription, translation, produce the protein. Here protein itself H plus ATPase, my friends. That H plus ATPase packed in the form of a vesicle. Vesicle fuse the Golgi complex. It undergo the modification. From the Golgi complex, every one of you look into the statement. From the Golgi complex, finally it will come and fuse to the membrane. Simple point. From the Golgi complex, it will go and fuse the membrane. The moment it will come and fuse the membrane, immediately what is happening, friends? The H plus ATPase pumps will get expressed. Then by using ATP hydrolysis, they pump the proton. The environment become acidic, activate the expansion. Once again expansion, loosen the cell wall. Simple, the cell is getting elongated. So I will summarize this. Listen. If you ask me how auxin is involved in the cell elongation, I can tell you that there were two hypotheses were derived to explain how the auxin is involved in the cell elongation. That is explained by two hypotheses. We call it as a activation hypothesis and synthesis hypothesis. According to the activation hypothesis, H plus ATPase pumps are already present. Even though they present already, my friends, they are not active. That's the point. Now how they are getting activated? By the auxin only. In second case, synthesis hypothesis, there will be a no presence of the H plus ATPase pumps only. Even during that particular time also, auxin only, friends, involved in the biosynthesis of H plus ATPase pumps. Auxin only helpful in the synthesis, auxin only helpful in the activation, auxin only helpful in their expression. This is how, my friends, auxin is involved in the cell elongation. Please kindly don't forget the statements. Now it is very important remembering the inhibitors. I am just reminding all of you one point, friends. Wherever you read inhibitors, promoters, stimulators, regulators, never ever miss it. I am not talking about only plant physiology or any other chapter. I am talking about all the 13 chapters in our entire CSIR syllabus. Wherever you heard the inhibitors of the auxin transport, all of you listen, friends. Immediately you have to remember. Very simple, friends. There are two type of carriers are there. There are two type of carriers are there which can bring the auxin inside and which can move the auxin from one cell to the another cell. What do you call it them, friends? One we call it as the influx carrier. One. Another one we called, my friends, efflux carrier. You know, friends, flux means movement. What, friends? Flux means movement. In means inside. Efflux. What do you mean by the word efflux, friends? Efflux means which send the auxin from one cell to the another cell. So if auxin wants to move inside, it has to enter into cell, it require influx carrier. If it wants to move from one cell to the another cell, it require efflux carrier. Friends, there are many type of inhibitors are there which can block the auxin efflux also, auxin influx also. Friends, here nothing much to remember. You people directly remember the name of the inhibitor. And anyway, listen one important statement. The auxin efflux carrier which send the auxin from one cell to the another cell, that efflux carriers, my friends, we can also call it as the pin proteins. What do you call it as, friends? Pin proteins. TBA, NPA, NOA. So what is TBA? Triiodobenzoic acid, naphthyl thalamic acid. My friends, please remember in a simple way, these two inhibitors, I will ask you in the next class also, remember, these two inhibitors, friends, which are going to block the auxin efflux carriers. Very simple point. Next one friends, NOA, naphthoxyacetic acid. This is the only type of inhibitor my friends which is going to block the auxin uptake. I mean to say which can block the auxin influx. Auxin influx can be blocked by NOA, remaining two can block the efflux. Now friends, finally what we have to read is the signaling. Signaling is one of the most important topic you have to read for every single hormone. I told you, friends, before 2015 December, in all the CSR papers, they never use it to raise the questions from the signaling at all. Every time they use it to raise the questions from the functions only. But friends, from the 2015 December, most of the time, the questions were asked from this auxin signaling. You take even 2020 paper also, there is a very simple question was asked from the receptors which are involved in the signaling. I will explain to all of you in a very simple, effective way. Only thing what you have to do is you have to understand with great patience. Listen to me carefully. Friends, the best method or the best way to read any hormone signaling is, you people has to remember 2 points. If you really understand the 2 points, you can answer any kind of a question coming from the signaling. You have to understand, my friends, what happens in the plant body in the presence of hormone. First point, what happens in the plant body in the presence of hormone. Second point, what happens in the plant body in the absence of hormone. What happened in the plant body in the presence of hormone, what happened in the plant body in the absence of hormone. My friends, these things you people has to remember compulsory. I will explain in a very simple way. Just listen to me carefully, friends. I will explain twice. Trust me, you will get a clear idea. Just listen to me. If auxin wants to active, or if the auxin want to show the response, there is an important transcription factor is required, friends. Remember this particular name of the transcription factor we called, friends, ARF, auxin response factor. Auxin response factor is the one which is basically involved in what purpose? Auxin response factor is the one which can show the response of auxin. Now all the students has to listen to me very carefully. When this particular auxin response factor, which is a transcription factor, can show their function, that is important. The ARF can show the action only in the presence of auxin. Sir, what do you mean by that? Which means if there is an absence of auxin, the ARF cannot show its action. If there is a presence of auxin, then only, friends, ARF is going to show their function. So all of you pay attention. Very simple, friends. In the absence of auxin, the transcription factor ARF, it become completely blocked. It become completely blocked by an inhibitor gene or early gene called as auxin IAA genes. Auxin IAA genes are called as early genes or the inhibitory genes, which can block the action of the ARF. Very simple point it is. Now hence there is no response. Listen to me carefully. But whenever there is a presence of auxin, friends, the concept is very clear. If the hormone auxin wants to show its action, first it has to attach us to the specific receptor. Without a specific receptor, auxin cannot show its action. Now the question comes here, what is the name of the receptor that is used by the auxin in order to show their mechanism of action? We called it as, friends, TIR1 receptor. TIR1 is the major receptor to which auxin will come and attaches. Now auxin will come and attaches to the TIR receptor. What should we do? We have to make the ARF become free from this inhibitory gene. You know, friends, for that what the auxin is doing? Auxin will activate the ubiquitin pathway. Every student pay attention. Auxin will stimulate the ubiquitin pathway. What is happening now? in the presence of ATP, ubiquitin molecule, you can see my friends, it will go and associated with this particular inhibitor or the early gene. Immediately what is happening, whichever get added by the ubiquitin molecule, listen to me friends, whichever added by the ubiquitin molecule, it can be degraded by 26S proteasomal complex. So 26S proteasomal complex, it is the degradative complex, will come and block this particular auxin IAA. It will degrade the auxin IAA. My friends, whenever there is a degradation of the auxin IAA, which will become free friends, ARF will become absolutely free. Now, you know friends, in the inactive form, ARF can exhibit as a one unit. But you only try to understand, in the active form, ARF become homodimer. Now this ARF will come and associated with the particular sequence in the DNA. What is the name of the sequence where the ARF will come and attaches? We can call it auxin RE, auxin response element. Very simple. Once the ARF attaches to the auxin response element, it will stimulate the growth and development of the plant. That is how the auxin can show the response. Here comes very interesting point. I told you many times, many times, if any hormone want to show the better action, all the hormones has to be released in a very low concentration only. More the concentration, less the response. Now you know friends, once the auxin response is over, what should we do? We have to control the auxin response. In order to control the auxin response, what we have to do? Once again, we will synthesize the auxin IAA genes. Now what this auxin IAA genes will do, my friends? Anyway, it will come and block the ARF once again after the response. Now whomever did not understand, my friends, I am repeating step by step once again, pay attention. In order to understand the signaling of any hormone, you have to remember what happens in the plant body in the presence of hormone, what happens in the plant body in the absence of the hormone. In the absence of auxin, the ARF, auxin responsive factor, which is a transcription factor, blocked by the auxin IAA inhibitory gene. Now whenever there is a presence of auxin, auxin will come and attaches to its specific receptor, which we called TIR1 receptor. When auxin comes and attaches to the TIR1 receptor, immediately auxin stimulate the ubiquitin pathway. Ubiquitin molecule with the help of ATP go and associated with the inhibitor gene. Then inhibitor gene can be degraded by the 26 proteasomal degradative complex. Concept is simple. Now which become free? ARF become free. Now the free ARF will convert into a homodimer. It will go and attaches to the specific sequence on the DNA. Then immediately auxin will mediate the growth and development in the plant body. Once the response is over, my friends, once again there is a synthesis of this particular early genes. What this particular early genes will do, friends? They will be basically involved in the production of ARF. They will be blocking the ARF to control the auxin activity. Friends, this is about the hormone auxin signaling and the entire plant hormone auxin. Now you all can ask all your doubts in the entire hormone auxin. Is it clear, friends? I feel it's clear for every one of you. You can ask me any kind of a doubt. Just say yes or no, friends, all of you people. All of you. Is that clear? Okay, I hope it's clear for all of you. Okay, so friends, listen to me carefully. I think in maximum 7 days, okay, in maximum 7 days, this unit 3 will be finished and unit 6 also will be finished. I am talking maximum. I will try to finish less than 7 days only. So you will be having a test. You will be having a test on the unit 3 also and unit 6 also. So please kindly prepare accordingly. Don't forget and don't neglect. And friends, in the month of April, I am going to finish 3 chapters at a time. I will finish unit 4 also. I will finish unit 7 also. I will decide what chapter we have to do, because unit 4 and unit 7 are little bigger topics. I will see what chapter I can finish, but I'll finish the 3 chapters. But as I told you friends, before June first week only I will try to finish 10 to 11 chapters in a very effective way with the help of more number of practice questions and PYQs. And I am requesting all of you people, please revise everything every single day without wasting time. Okay, so friends, after 10:30 a.m. you check the website. We will upload unit 3 practice questions and unit 6 practice questions also. So please download the practice questions of unit 3 and unit 6 and try to read it. Okay, try to practice. Check after 10:30 a.m. Okay friends, that's all for now. We will meet in the tomorrow morning class. We will continue the post transcriptional process. Friends, less than 7 days I'll finish the 2 chapters in a very effective way. Okay friends, that's all for now. Thank you so much. Thank you for your all patience. Thank you. Have a good day to all of you. I'll see you in the tomorrow class. Thank you. And last and final thing, in the month of April, I will be taking every day 3 hours of classes. I will make a simple schedule. I will tell you how I will take the 3 hours. By that we can finish the most of the syllabus. Okay friends, thank you all. Bye bye.